if we have an organism like Spirogyra, so in Spirogyra we have a repeated body design. After some times, we see that there is a repetition of same body design is taking place. Okay, like this is Spirogyra. So this the pattern start repeating. Okay, then this one, the same body design repeat after a regular interval. Okay, and this is called fragments. This is called fragments. Later, when the Spirogyra mature, these fragments are separated. These are called fragments, and these are separated, okay, and behave as new organism. Okay, so Spirogyra is a freshwater living multicellular organism which have a repeated body design after regular intervals. So these are separated out. These fragments are separated out and behave as new organism. Behave each fragment behave as new organism. Okay, you understood? So what is fragmentation? Tell me. Tell me what is fragmentation. So note it down. In some organism like Spirogyra, water is, we can say that when it get mature, there is a repetition of same body designs. When Spirogyra get mature, there is a repetition of same body designs take place. And this regular repetition is called, or we can say that this repeated body design is called fragments. Okay. Okay. And after some days or after some time, these fragments get detached and behave as new organism. Please note it down. Note it down. Done?
Noted, sir.
Okay.
Thanks, sir.
Okay. So let's move further. Shweta, done?
Yes, sir.
So now moving to the next part is vegetative propagation.
Yes,
Now we have studied about the reproduction in animals like spore formation. Okay, we have studied regeneration. Regeneration is not a sexual method of reproduction. It is a developmental process. Okay, it is not an asexual method of reproduction. Budding we have done. Okay, and we have done the uh, along with this we have done various types of uh, methods of reproduction like budding, binary fusion, multiple fusions. Okay, fragmentation, regeneration. Okay, budding. We have done this. Now we are just talking about these. These were the previous one was the reproduction, asexual method of reproduction in animals. Okay, now today we're going to discuss the asexual method of reproduction in plants. Okay, so asexual method of reproduction, asexual method of reproduction in plants is called vegetative propagation. Is called vegetative is called vegetative propagation. Propagation. Okay, and what is vegetative propagation? Tell me, what is vegetative propagation? So we can simply say that vegetative propagation is the method of producing new plants. Okay, vegetative propagation is the method of producing new plants in which we are able to produce new plants by their vegetative parts, by not using their reproductive organs. Okay, so if I'm saying that, what are the vegetative part of the plants? So vegetative part are like root, stem, leaves, etc. And reproductive part is flower and seed. Okay, so we are not going to use this one. These are prohibited. We are not using. We are just only going to use root, stem, okay, stem and leaves. Only we are going to use these three. Either we will use root, either we will use stem, either we will use leaves for growing new plants. Okay, you understood? Understood?
Yes, sir. Yes, sir.
So here we will say that vegetative methods.
Yes, sir.
We can say that vegetative propagation is a asexual method of reproduction in plants in which we can grow new plants by using the vegetative parts, okay, of the plants like root, stems, leaves, or we can say without using their reproductive parts like flowers and stem. Okay, so noted down the definition. Vegetative propagation is an asexual method of growing new plants from their vegetative part like stem, root, leaves without involvement of their reproductive organs.
Noted, sir.
Noted, everybody?
No, sir.
Noted,
Yes, sir.
noted, noted. And yes,
Done,
here
sir.
I'm just talking about Shweta. Shweta was present in the previous class. Abir was
Yes.
also present. But Simon, Tejasvi, you didn't take the previous class of this chapter. You were absent in
No,
the
sir, I
previous
was
class.
present, sir. I was there.
You was there, Tejasvi.
Yes, sir, I was there.
Okay, you studied regeneration. So
Yes, sir.
Abir, do you take the previous class and Shweta?
Yes, sir, I took the previous class.
Okay, so Abir, Shweta, Tejasvi, Simon. Okay, let's move further. Done? Noted? Noted?
Yes,
Yes, sir, noted.
Now this vegetative propagation can be of various type. Vegetative propagation is of two types. Okay, one is the artificial, and another we can say that one is natural, and the another one is vegetative. Sorry, artificial. The another one is artificial. So what does it means? That there are some plants part, or we can say vegetative parts of plant like stem, root and leaves, which has the capability that when they buried under the soil, they can give rise to new plants. Okay, for example, if I'm just talking about in natural methods, like eye of potatoes have some bud like structures on it, and when you cut the bud and you buried it into the soil, then new plants can arise from this one. Okay, so eye of potato. Okay, similarly, the we can say that the nodes of gingers have some nodes like this. Ginger is like this, so it have some scaly leaves over it, and they have some nodes like structure. You can see that one if you have seen the ginger. Okay, so this is some nodes like structure. There's some at some place. So when these nodes are buried under the soil, then they can give rise to new plants. Okay, similarly, the roots of carrot, they can also give rise to new plants. So, and the leaves of bryophyllum leaves have some notches on its surface. Okay, it is just like this. Okay, so they have notches over it. So when they buried under soil, it can produce new plants because it can develop adventitious roots, which can give rise to new plants through it. Okay, so when they are buried, so this is the leaves of bryophyllum, leaf leaf of bryophyllum can give rise to new plants. So we can talk about this one, that they are different natural vegetative parts which has the capability to give rise to new plants. Okay, so we can grow new plants by just using their vegetative part. Okay, so let's talk about this one. So there are the two types of methods. One is the natural method, and another one is the artificial method. Okay, so we can say that leaf of bryophyllum, first of all, this we are talking about the natural methods. Okay, different types of natural methods. First one is leaves of bryophyllum. So leaf of bryophyllums have notches on the surface from which adventitious buds arises. When, like, this is the leaves of bryophyllum. Okay, and when you buried it under the soil, yeah, it get buried under soils. So we can say that adventitious buds arises, which when buried under soils, and has this bud has the capability to give rise to new plants. So we can grow new plant just uh, by soiling, or we can say that by sowing the leaves of bryophyllums. Okay, and not every plant's leaf has this capability. There are some plants specifically that has their leaves has capability to give rise to new plants. But the question comes that why does we can grow the plants with bryophyllum leaves but not with jasmine leaves? The simple answer is because jasmine leaves do not have adventitious buds or don't have notches, which when buried under soil can't give the new buds. Okay, so while they in bryophyllum, we can have such type of bud which when they are buried under soil, they can give rise to new plants. So please note it down. Note it down. I'm going to show you the leaves of bryophyllum then.
Done, sir. Noted, sir.
Okay, okay.
Done, sir.
So we can see that these are the leaves of bryophyllum that you can see here. That these are the leaves of bryophyllum. Okay, and they have notches over it. When they buried under the soil, they can give rise to new plants. Okay, so these are the leaves of bryophyllum. Okay, got it? They have notches. When they are buried under the soil, then which produce buds. Okay, we can say that these notches produce buds. There you can see without. When these buds are buried under soil, then they can give rise to new plants. You understood? Understood?
Yes, sir.
The bryophyllums have capability to give rise to new plants. Okay. Similarly, there are the other options also. Like we have eye of potato. Okay. So eye of potato also have we can say that they have notches. Okay. They have points. Like I'm going to show you the eye of potato also. So you can check it here that these are the eyes of potatoes. Okay. So when they are buried under the soil, like you see that this one, you can also see that when you keep the uh, potato for a long time, then these buds by self arises. Okay. and we can say that these are capability to give rise to new plants. Okay, got it? If you kept the
Yes, sir.
onion also for long time, then this onion can give rise to new plants. You can see that if you keep the onion, then I from the onion, the green colors pigments or green color leaves arises. Okay, so these are called the eyes of potato. Okay, so please note it down that potato is just a modified stem. Okay, sweet potato is modified root, and normal wala potato is just a modified stem which have notches on it, on its surface, called eye of potato. Potato, when buried under the soils, okay, it it has capability to give rise to new plants. So what is it? It means eye of potato. Okay, so the eye of potato has means the has capability to give rise to new plants. Please note it down. When you're done, please let me know. Okay.
Done, sir.
Done. Everyone in the
Done,
class.
sir.
Done, sir.
Similarly, nodes and internodes of gingers. Nodes and internodes of ginger. Nodes. So you can check that this ginger is also a modified stem. Okay, ginger is also a modified stems, and you can check it. This is ginger, and this is the bud. You have seen this one at your home also, that gingers have bud. Then when you peel the ginger, you find that when you are peeling the this bud, this is fleshy like a structure. This is not rough. Here, if you peel it, you will find some roughy area, but you will find this bud. When you are peeling this one, you will find some slippery like structure. This is called bud. When this bud is dried, or we can say buried under soil, it can give rise to new plants. So these birds have the capability, these birds that you have seen in the gingers. Okay, these birds have the capability to give rise to new plants, and ginger is a modified stem. Okay, you understood? Understood?
Yes, sir.
Yes, sir.
So we can say that here. And similarly, the bulbous of agave, there are so many vegetative parts of the we can say that, of the plants, different plants. Okay, that has the capability to give rise to new plants. So bulbous of agave. So we can say that this is a plant, or you can check it that this is the bulbous of agave. So agave is the plant. Okay, and these are the bulb like structures. So different plants have different types of structure that has the capability, like you can check it here, that these are the bulb like structures. Okay, when these are fallen from this one, it can give rise to new plants. So new plants can be grown not only by using the seeds and flowers. It can be grown by the vegetative part of the plant. Okay, got it? But not every part of every plant have their every vegetable organs reproductively active. Okay, so we cannot say that. We can say simply that some plants vegetative parts are reproductively active. The leaves of jasmine is not vegetatively active, but we can say that the leaves of leaves of bryophyllum is vegetatively active. Okay, you understood? The next type of method is called artificial methods. In artificial methods, of vegetative propagations, what we does actually. So in artificial methods of propagations, we cut different parts. We use different techniques to grow new plants. For example, this is cutting. Okay, cutting. So cutting of stems, root, and leaves. We can simply say that when you are cutting the stems, roots, and leaves, the new plants can be grown by this method. Okay, got it? Got it? So different in stem cuttings, root cuttings, and leaves cutting. They are the different parts. If we are cutting the stems, stem cutting is very common in China rose, sugarcane. You cut it and you bury the stem into the soil, then you will get a new plants. Okay. Similarly, root cutting, if you cut the root of lemon, you can get the new plants. And leaf cutting, if you cut the leaves of sansevieria plants, okay, then it can give rise to new plants. Okay. Understood, everybody?
Yes.
Yes, sir.
Yes, sir.
So cutting is the method in which suitable part of the plants like stems, root, and leaves is being cut, and then they have the capability to give when they are buried under soil. We just, if we are just using stem cutting, then we just choose the stem or that part of the stem which is going to have much branches. Okay, much leaves. Okay, so if I'm talking about like rose plants, cutting in rose plant, cutting in rose plant. So you can check that when you cut the rose plants, okay, and these are being cut. So rose plants are cut here, okay, and they are just producing their roots. Okay, so these are just cutting, and when they are cut like this way, on the day one this is showing you, and on the 18th day this is going to give you the new leaves. So we just cut it like this one, like this way, V shape, and then we buried it under the soil, then it can give rise to new plants. Okay, got it? Got it, everybody?
Yes, sir.
Yes,
We
sir.
just cut it at 45 minutes. We are just making the 45 angles. We are not just cutting it like, and moreover, we just take the part, we just choose that part of the plant which is going to have very much leaves. Okay, leafy part. Okay, and next is we use stem cutting in lemon. Sorry, root cutting in lemon. So root cutting in lemon. So when we cut the root in the lemons, then we can give rise to new plants. So you cut the root of lemon, and the new plants can be grown by this one. Okay, lemon tree and leaves of sansevieria. Okay, so leaves of sansevieria plants, you can check it on Google. So this is also used for giving and rise to new plants. Okay, so please note this one, cutting the artificial method. Note this one.
Sir, what is the example for leaf cutting?
Leaf cutting. Leaf में this is leaves, and this is sansevieria plant. Sansevieria plant. Have you seen sansevieria plant? You must have seen that one, but you don't. I think you don't remember that this is sansevieria plant. So this plant is very rich in. Okay, this plant is very rich in producing oxygen. and this can easily be grown in the uh, low lighting conditions. So we just keep this plant in the home, okay, to produce more much oxygen. Okay, so this Sansevieria plant is very important for producing oxygen. Okay, got it? So this, if you cut the leaves of this one and if you bury it in the soil, then this Sansevieria plant can give rise to new plants. Okay, got it?
Yes, sir.
Yes, sir.
Noted this one. Note it down. When you're done, please let me know. Okay. Done. Noted.
Yes, sir.
Why are you not replying?
Done, sir.
The next method is called layering. Layering is a method in which we just bend the plant part, okay, and buried it under the soil. Okay, like we are having this one. Like we have some plant, okay, and it have some branches, okay, and just we take one of the branch and buried it under the soil. Okay, it is generally done in the climbing plants, okay, not in the strong plants. And like jasmine can be grown by this method. Okay, so layering, we just layer this part, and later we will find that this layer part will start growing new plants, new roots, and new plant will start growing from this one, and later we detach from this one. Okay, so this is called layering. This is called layering. So if we talk about this one, so this is called layering. Okay, so layering, we can say that in layering, a twigs or a branch of plant is bent down below the soil. This is called layer. This bent part is called layer. Soon, this portion develop new roots and give rise to new plants. Later, the parent part branch is cut and deattached. Okay, so please note it down. Then I will show you that how layering is done. Noted?
No, sir.
Noted.
Noted, sir.
Noted.
Done, sir.
Okay, so this is the type of structure that you see that in the layering. So in layering, we have this plant part and we just bend it down. Okay, and then new plant grows through it, and later this part is detached. Okay, got it? We will say that after some time, this part will get detached and we will be able to see the new plants. Okay, so this is called layering. Layering is also done in the air also. Means it is not necessary that we are going to draw layer this one. Sometimes this portion, or we can say that this portion is cut here, and a cow dung and a tape of means some humus is bent over it, or we can say wrapped over it. And after some time, we find that this part start growing new roots. Okay, and then this part is cut because it now this part is going to have root, and then we can bury it under the soil. Okay, and then this part will act as a new plant. So let me show you that one also, that how air layering is done. Air layering, that means we can say that budding is done, sorry, layering is done in two ways. One is air layering, another one is ground layering. Okay, got it? Got it?
Yes, sir.
So let me show you air layering. So you can check it here that this is the plant where we can uh, first we peel it out part of the plant. Okay, and after peeling we can do this one. Okay, and we just put some artificials cytokinin or some means some cell division pigments, sorry, cell divisions hormones, cell dividing hormones, and then we wrap it with some clay. Okay, like clay, and just wrap it with the, okay, like this. Soil with humus enriched in, provided from infection. We just tied it with the polythene. Okay, and then after some days, we will remove this polythene and we'll find that the new branches are arrived here. Okay, so you can check this method also, and this is another method for growing new plants. The 35th day, it is showing like this. Okay, after 35 days. So it has been removed here. Okay, and then new roots arises. Okay, so you can cut it from this portion. You can cut it and then you can grow this part because now it is having branches. Now this, so along with this, you cut it from here. Okay, and you can bury this plant under the soil. Now this plant has capability to give rise to new plant because it has developed new roots. Okay, got it? Inside this clay or inside this soil, there is new roots. Okay, got it? So this is the way
Yes,
that
sir.
how can we grow new plants. So this is removing this, and you can bury this one like this way, and you can grow new plants. Okay, so this is the method of air layering. Okay, the next type of method we called it is the next type of method we called it is grafting. Grafting is another method in which we are just in the sexual method of asexual method of reproduction. We just use only one characteristics of the plant. We are just going to have only one parent. But in grafting, what we are going to do here is we are just going to join the two plants to different plants. Okay, like we are having this plant first. Okay, and we just cut it like this, and then a new plant is cut like this way. Okay, new plant, we cut it like this way. Okay, and then this is put over it, and then it will gives you the new plant. Okay, so these are put over it, and then we are going to wrap it, okay, with vaccine tape, waxing, or we can say that grafting wax. Grafting wax we are applying, and we are tidying this one, and then new plants means this plant is going to have means the characteristics of both the plants. For example, this is the basal part, which is of sweet mango, and this is a basal top part, which is going to have means it is it bear this plant bears so sour mangoes. Okay, khatte. So both the characteristics of sweet and sour are going to get in this tree, or then the newly grown tree is going to have the fruit of both the characteristics. So this is the asexual method of reproduction, but in this we are just using two different plants and their characteristics desirable to obtain the plants with desirable characteristics. Okay, got it? So this is called grafting. Okay, what we called it? We called it grafting. You understood?
Yes, sir.
Yes,
Okay.
sir.
So we can check it here also. Wait a minute. Grafting. So I'm going to show you here also. So you can check it, dear students, that this is the plant that we peel it first like this way. Okay, and then and the same thickness we are going to have the plant. You don't think that we are just going to use a bigger plant or with a very thicker stem. No, we just take the plant which is not very thick in the initial stages. This is the mango tree. Okay, mango tree, and we just peel it from this one in the V shape. You can peel it. Okay, and then these two are tied together such that they are both the xylem, both the top part and the bottom part, xylem and phloem tissue, any vascular bundles match up. Okay, the food and water conducting tissue can overlap each other. Okay, of the top part and the part and the lower part. Okay, and then we tied it. We just put it here. Okay, then we just put it here, and then we will apply the grafting wax. And the top part is called secant, and the lower part is called basal. Okay, we just arrive it like this, and we just wrap it with the polythene or some grafting wax is applied to prevent it from we can simply say that to prevent it from infection. And after some days we will check that this secant, this top part also start producing new plants. Okay, means it will start growing and develop. You can see that here. After some days, this top part will get attached with the basal part, and the new plant, this part will start growing further. Okay, got it, everybody in the class?
Yes, sir.
Yes, sir.
Yes, sir.
So you can check here that this is the top part which was connected, and this is bearing leaves now, and initially it was not bearing leaves. So you can check that this is the plant having different colors of leaves, because this plant is going to have the characteristics of both the parts. The lower part is going to have only green leaves. You can check it here. Okay, and the top part is going to have the leaves of both the colors, because this top part is going to get the characteristics of both the plants. So this is how we can simply say that we use uh, grafting. Grafting is an artificial method. Okay, So please note it down. Here, let me show you. That grafting is another method. So note it down. Have you noted the layering?
Yes, sir.
Okay, so what happens? Let me show you that in this technique, it is a technique of joining two plants in order to obtain the desirable characteristics of both the plants. Okay, in this method, a plant is cut from the stem. Lower part is called stalk, and the surmounted. This is called stalk, and the surmounted part is called secant. Okay, such that both the stalks and vascular bundles should overcome each other. Means they should overlap each other. You can write it overcome or overlap each other. Okay, and they must be of same thickness. Means they must be of same thickness. You cannot say that a plant secant is very thin and stalk is very thick. They are, must be both secant and stalk must be of same thickness. Okay. Okay. Now stalk and secants are tied together and a grafting wax is applied in order to prevent infection. Okay. And then this plant start producing. Okay. Note it down. Grafting. Done. Noted, dear students.
No, sir.
Noted fast.
Done, sir.
Done,
Done?
sir.
Okay. Now there is another methods. We call it micropropagation. Micropropagation is called tissue culture. Okay. Let me show you that what tissue culture is. In the ancient times, tissue cultures was used, or we can say that it was not in the ancient time. It is a modern technique also. We just extract a new plants. Let me show you that what actually we does. We just, for example, this is a plant, okay, and we just extract a part of a plant, a tissue, a cell or a group of cell. We just extract it, okay, and we just take this part that we have extracted and we just grow it artificially in the nutrient rich medium, with along with some growth promoting hormone like oxygen and cytokinin. What I'm saying, let you please listen one more time. We just take the tissue or a group of cell. For example, this is a plant. We just take the part of this plant, okay, and then we grow it in nutrient rich medium. Okay, what we does, we grow it into nutrient rich medium along with some hormone that promotes cell division and like oxygen and cytokinin. Okay, later this cell that we have taken here, it start multiplying, it start growing. Okay, and from 2 to 3, then it form a big mass of cell, we called it callus. Then it form a big mass of cell called callus, and this callus when buried under the soil, okay, then it can gives a new plants. Okay, so this callus can give rise to new plants when buried under the soil. You understood? So this is called micropropagation and it is also called tissue culture. Tissue culture. You understood? What is micropropagation? What is tissue culture?
Yes. Yes, sir.
So let me show you this one also. Tissue culture technique. So initially we take the suitable part of the plant. Okay. Like you can check first. Okay. Here. So we just take the cell. Okay. And this was just, for example, we just take the part of the plant. Let me show you here. Okay, and then we start growing it in a nutrient rich medium. Okay, we just take the tissue part from this one and then we start growing it in the nutrient rich medium, and then after some times it becomes we can say that when the nutrient rich mediums are provided, then it start giving rise to it becomes a big mass of cell which has the capability to give rise to new plants. Okay, so tissue culture is a special technique in which we take a tissue or a group of cell that is called explant. What we call it? Explants. Okay, the tissue that we take from it, we called it explant, and explants when buried under the soil. So when we grow it in the nutrient rich medium, then it start multiplying along with some growth promoting hormone like oxygen and cytokinin. Okay, you can see here. Then we just produce it like this. We take the tissue, like leaves, we cut the leaves, okay, and we just take the 4 part of these leaves, okay, and then we start growing it in the nutrient rich medium. Okay, like sucrose, in organic material, vitamins. Okay, so then after some times this becomes callus. Okay, big mass of cell. These are differentiated and these are growing a big mass of cells. Now when this big mass of cells is buried under the soil, then it can give rise to new plants. So you just put it along into 4 different parts, these 4 leaves and then you, okay, and then when you bury it under soil, the new plants can be grown by this method. Okay, you understood? So just a part of leaf, we take a leaf, we just cut into 4 parts and new 4 plants can be grown. So can you imagine that how many thousands of plants can be grown by a single plants? Okay, with same characteristics. The same characteristics plants is, this is the benefit of this technique, that identical plants of same height, because only one plant's DNA is being used here. So we can say that in this method, we can say that the plants with same desirable characteristics can be grown by this method. Okay, and those plants which does not grow, which don't bear seed, in those plants we have predominantly this method for growing new plants. Okay, so and this is a safer method because this is a disease-free method because we are just extracting the plant. Okay, and we are just growing the plants in the fresh medium where there is no chances of infection. Okay, so the initial chances of infections generally when seed is buried in the soil, the seed get infected. But here, because we are just growing the plants in the nutrient rich medium first of all, and then we are just transferring this big mass of cell into soil. Okay, got it? So what is tissue culture? You understood that we just take the plant part, okay, or leaves, that is called explant, and then we just transferred it into the nutrient medium. Then this nutrient medium start growing new plants. Okay, means it start growing big mass of cell. Then this big mass of cells when transferred to this soil, this is called scalus, and when this is transferred to the soil, then it gives rise to new plants. Okay, you understood?
Yes, sir.
So let's please take the screenshot of this one, and you have to do this one in the homework. Okay, so please take this one, and later you have to do this one. Okay, note it down. Okay, wait a minute. It is an advanced methods of reproduction in which large number of small plantlets can be obtained by using this technique.